All right, folks, today we're going to be talking about the cell cycle and mitosis. So this is going to help you out if you were a little bit stuck in some of the questions from last week. And then I'm going to make a video about meiosis as well. But first, let's talk about the cell cycle. So the cell cycle is a cycle of growth and replication that cells undergo. And we're talking about eukaryotic cells. Those are the cells that have a nucleus. So this is the kind of thing that's happening in human cells. It's important to note that not all cells in an organism, like a human being, operate on the same schedule. They don't all replicate at the same speed. Many cells in a complicated organism like a human being replicate incredibly slowly and sometimes not at all. There are certain neurons that actually they exist after they're set up. They just exist in G1. That's the growth phase where they're just doing their job, and they exist in that until they die. Some neurons can replicate, but we saw that there's a very limited ability for those cells to make copies of themselves, and that's why brain injuries are a big deal, because you don't get a lot of those neurons back. Anyhow, so this is the cell cycle. It starts in G1, so we start actually right here, and G1 is what we call the growth phase. That's the cell doing its job. So if you are a neuron, you're doing neuron stuff. If you're a liver cell, you're doing whatever chemical reactions are demanded of you at that time. Whatever. So the cell's just doing its thing. When a cell gets big enough, or enough time has passed, or it's been given instructions to get ready to divide, it starts down a path called cell division, and we're going to talk about that. And the first thing that you have to do is, if you want to make two cells out of one cell, there's a lot of stuff you need to get in order. You got to get all your ducks in a row, so to speak. So one thing that you need to do is, if you want both of your daughter cells to have exactly the same amount of DNA, because they have to have the exact same amount and type of DNA, you got to copy the DNA. And so S phase is the synthesis phase. This is when we copy DNA. And as you could imagine, a lot of things can go wrong in here. The cell has ways of kind of figuring out if something's gone wrong. It's got ways of getting back on track if there's a problem. And if the cell is unable to get back on track here, it has ways of preventing itself from copying. Because if you think about it, if your DNA is all screwed up, you don't want to make a copy of that cell. Because if your DNA is messed up, there could be mutations, there could be breaks in the DNA, there could be a whole bunch of disastrous things that could be happening. So if you can't fix the problems, what happens is, if there's ever a major malfunction in S or G2 or mitosis, the cell will it'll self-destruct. It's better to lose a couple of cells than to have weird, mutated, DNA damaged cells carrying on. So we've copied our DNA, and you can see this takes a long time. It's very it's a work intensive and energy intensive process, because in a human being's cell, in all of the cells in your body, other than sperm and egg cells, and we'll talk about those later, you have a full set of DNA. And for a human being, that's about 3.2 billion letters worth of DNA. So DNA, the four basic letters are A, C, T, and G. We'll talk about that in the next chapter. But suffice it to say, 3.2 billion letters is a lot to copy. And you have to copy it with 99 point and then 6 9s. So 99.999999% accuracy. We can't get to 100, but that's a story for another day. So once we've accomplished our DNA synthesis, now we have 2 copies of every chromosome. That makes sense again, because remember, we want to make sure that that both of the new cells we make has 1 copy of each chromosome. For human beings, we have 23 pairs, that's 46 chromosomes. So that means that for a brief moment during this phase, we won't have 46, we'll actually have 92. And then we'll separate them and we'll give each cell 46, aka 23 pairs. Then we have gap 2 phase. This is, or it's called gap 2, but we're calling it growth here. Growth phase 2, you are making sure that you have enough organelles. So you got to make sure that both cells get some Golgi. You got to make sure both cells get some rough endoplasmic reticulum. You got to make sure both cells get a whole bunch of mitochondria and lysosomes, and they both get some nucleolus, and they both, you need to make sure that you have enough for both of the daughter cells. Because if you don't, what are you doing mitosis for? Why are you trying to copy your cell if you don't have enough of each type of organelle? So once you're done copying all your organelles, then you commit to mitosis. There are also checkpoints in here where if something goes horribly wrong, the cell can basically either try to fix it or give up and just pop, self-destruct. So let's talk about the way that DNA is organized inside of cells. When a cell is doing its life cycle, in G1, remember G1 is when the cell is doing its thing. Since the cell is doing its thing, your DNA is all uncoiled. It's not packed up into these nice little butterfly-looking packages. No. Since you are doing cell stuff, you need to read the DNA. And if you are reading DNA, you can see here there's some uncoiled DNA. It's even more uncoiled. This is DNA that is actually being read. So that means that the picture here, which is an electron microscope image where we insta-killed the cell, used chemicals to freeze it where it was, and then we took a picture by bouncing electrons off of the nucleus of that cell. This cell, when it was killed, was doing some work. It was actually reading DNA. So these little loops that you see, these are parts of a chromosome that are open. Just like when you open a book, you can read it, but you can't read a book when it's closed. So these are actually open reading frames. These are parts of the DNA that are being read. So if this was a living cell, cells make proteins. That's one of the most important things that they do. They make different kinds of cells, make different kinds of proteins to stay alive, to contribute to the well-being of the cell, and therefore to the well-being of the whole organism. And so you get the instructions on how to make each type of protein from your DNA. So these are being read. So this is called chromatin. It's basically just unspooled, unwound, unpacked DNA. We cannot see individual chromosomes here. You're going to see this in G1. Remember, that's the cell doing its stuff. S phase, when you're copying DNA. And also G2 phase, which is the rest of the preparation for mitosis. When you are committed to doing mitosis, one of the things that you got to do is pack your DNA up. Just like when you move as a human being, you go from one house to the other. You don't just take individual items. I mean, maybe when you guys are like 19 or 20 or 21, you're moving into your first place. Yeah, you might just slam a whole bunch of your stuff into your trunk and into the back of your car and into the back of your buddy's car. But the smart way to do it is to actually suck it up and buy the boxes and put all of your stuff in the boxes. Like go to U-Haul, get some boxes, because it's going to make transporting your stuff way easier, and it's going to mean that you don't lose things. So what cells do is they package their DNA. They only package it when they're gonna do mitosis or meiosis. And they get packaged into these little lines or little X-shaped structures. This single line with the bulge in the middle is called a chromatid. It's a single chromosome that's been packed up. You see this in anaphase and telophase of mitosis, which we'll get to. So this is a single copy of a single chromosome. In the center, we have a bulge called a centromere. This is going to be a very, very important feature when we talk about mitosis. It is an attachment point for a spindle fiber. And we'll talk about that in a second. So underneath we have sister chromatids, or duplicated chromosomes. Duplicated chromosome meaning that this is two chromosomes. They can be pulled apart and each one can go to the daughter cells. But these are genetically identical. So that means that if I were to slice this chromosome right down the middle, we would end up with two of these. And they're perfectly identical to each other. So that means that this one, A, and this one, B, they are perfectly identical. So one daughter cell will get the copy called A, one will get the copy called B. We see this in prophase and metaphase. So again, these are sisters because they are identical, 100% identical. They are a copy of one another. And then we have homologous chromosomes. Because most organisms are sexually reproducing, not all of them, but since they're mostly sexually reproducing, that means you have a copy of each chromosome from your mom, and you have a copy of each chromosome from your dad. We call that a 2N organism, or a diploid organism. In human beings, we have 46 total chromosomes, one copy of each from each parent. So that means you have 23 chromosomes that you get from your mom and 23 chromosomes that you get from your dad. Chromosomes 1 to 22 are called autosomes, and then chromosome number 23 is a sex chromosome, which we've kind of talked about already. So you have a copy of 1 from mom, a copy of 1 from dad, a copy of 2 from mom and 2 from dad, until all the way until 22. Then you have 1 sex chromosome from mom and 1 sex chromosome from dad. You get an X chromosome from your mom, and you get either an X or a Y from your dad. I want to say this because there's always some student who completely doesn't get it. These are shaped like an X, but they're not X chromosomes. These could be any of the 1 to 22, or they could be the X chromosome. This is just a copied, duplicated version of a chromosome. So homologous chromosomes are chromosomes that have the same genes on them, but one of them is from your mom and one of them is from your dad. So that means this could be chromosome number 1 from mom and 1 from dad, or it could be chromosome number 2 from mom and 2 from dad. They are the same, they contain the same genetic material, but they might be slightly different because your mom and your dad are different people. That means that chromosome number 1 that you get from dad is not the same as chromosome number 1 that you get from mom. So AB is homologous to CD. So they are similar, but individually different. So let's jump into mitosis. So what's the point of mitosis? It's really simple. It's to make 2 copies of a cell. The point of this is to make 2 identical copies, and we call them daughter cells. And there's the 2 major reasons that you'd want to do this is if you want to make your organism bigger. If you count the number of cells in a baby versus the number of cells in an adult, there's a different number of cells. An adult has way more cells. Where did they come from? Mitosis. The other reason that you might want to do this is to replace dead cells. If you accidentally, you're cutting some broccoli or some cauliflower and you accidentally nick your finger, you've got an injury. You just killed a whole bunch of cells, like thousands of cells possibly, even from a minor cut. And you gotta replace them. You can't just, not replace dead cells. You need to replace them in order to heal. So what will happen is around the injury, surviving cells that are nearby will basically catch wind. They will be aware. They'll find out that some cells beside them have died. So if you're a skin cell and your neighbor skin cell just got sliced by a paring knife or by a kitchen knife, the dying cell basically gives off chemical signals that's like, oh, I'm dead, you gotta replace me. And then the cell beside it will be like, all right, I'm gonna replace you. I'm gonna do mitosis and make a copy of myself to replace my neighbor that just got killed by the kitchen knife while we were chopping vegetables. So this cell that I'm going to use here is a very simple cell. It would be called 2n equals 2. So that's 2 parents, 2 chromosomes total. In other words, 1 copy from mom and 1 copy from dad. So these are homologous. First thing we do is S phase. We copy all of our DNA. You can see I drew a lot more red squiggle and a lot more blue squiggle. So the DNA has been copied, but it has not been packaged yet. So it is still chromatin. Once we reach the end of S phase, it's time to do mitosis proper. So we're skipping G2. Suffice it to say, all the other stuff in the cell gets copied in G2. We need to make sure we have enough organelles. So first phase of mitosis proper is prophase. A lot of stuff going on in prophase. You'll notice we are not blue squigglies and red squigglies anymore. We have packaged our DNA up into chromosomes. Remember, the blue one in this case is homologous to the red one. And also remember, in our destination, at the end of the day, we should have 2 cells that look exactly like this. That means that each of our daughters should have a red chromosome and a blue chromosome. And they should only have 1 copy of each, not 2 copies of each. So in prophase, we package our chromosomes up and we bundle them up into structures that we can see. We also start to break down our nuclear membrane. So the membrane that holds the nucleus of the cell, it actually disintegrates. It's a temporary thing, and the reason why it needs to disintegrate is because these very cool structures called centrosomes. I know there's a lot of centro words. These centrosomes are going to basically shoot little tiny grappling hooks, and those grappling hooks are going to connect to the centromeres of the chromosomes. The reason why is because these spindle fibers are going to be used to move those chromosomes around. And these spindle fibers are actually made of something that you know about all the way back to chapter 10 of Bio 20. They're made out of microtubules. So they're made out of structural proteins that will connect. And so once these have connected, that's the end of prophase. There are really two subdivisions called early prophase and late prophase. In early prophase, you can still see a little bit of the nuclear membrane as it dissolves. By late prophase, all of our spindle fibers are hooked up and we're ready to roll. So let's go on to metaphase. Metaphase is the middle phase. It's the second part of the acronym PMAT, which is prophase, metaphase, anaphase, telophase. In metaphase, we have our cool little structures, the centrioles or centrosomes. They are using their little grappling hook microtubules, the spindle fibers, to move the chromosomes around. And they move the chromosomes around onto this imaginary equator line. Just like there's no like real equator, there's no like, if you go to the equator of the planet, there's no line on it like you see on a globe or a map. It's an imaginary line that we invented to make sense of the globe. Same deal here. This is an imaginary line called the metaphasic or metaphase plate. It's not an actual thing, it's an imaginary line. But you can see we have our blue chromosome and the homologous red chromosome lined up in the middle. Here's a picture of some real cells. I believe these are cells from a whitefish. And I know we don't have the resolution to really see individual chromosomes in here, but these black or dark stained pieces, these are these doubled up, these copied chromosomes. You can also see in reddish the microtubules known as spindle fibers. So these have lined everything up, and now we're going to start pulling. This is not an animal cell this time. This is actually a plant cell, and the reason why, if you remember in Science 10, we always used to draw animal cells as kind of a round blobby shape, and we always drew plant cells as kind of a rectangle or a square. This is kind of why. So here we can actually see in dark purple, these are sister chromatids being pulled apart. So for example, we could say that this is the red one, and this is the sister of the red one. So it's also red. And then we could say that this one is the blue one. So one of the sister chromatids of the blue one, and then this one is the other sister chromatid of the blue one. So these microtubules are being basically shortened, and when they shorten, they reel in the chromatids. So again, that means that this chromatid is going this way, and that chromatid's going that way too, and then these chromatids are going towards the opposite end of the cell. So we split them apart, and this is cool because this means that after we divide the cell on the right will have a red copy and a blue copy, which is what we started with. And the left cell will have a red copy and a blue copy. Everything's beautiful. Now we go to telophase. Oh, one last thing I would like to say. Ana means apart. You might remember this from anabolism, which is the breaking down of big molecules into small molecules. Ana meaning apart, so breaking apart. So anaphase is the breaking apart of these doubled up chromosomes and the pulling of each piece of the doubled up chromosome towards the pole. And then we have telophase. Telo literally just meaning the end. So in the end, we've already pulled all of our sister chromatids apart. So they're where they need to be. So we can start putting together our nuclear membranes again. So that means that the cell on the right gets its own nucleus with the right number and right type of each chromosome. And the left cell gets its own nucleus with the right number and right type of chromosomes. So the nuclear membrane's reforming. We have a cleavage. In other words, this is going to start splitting down the middle. What's really happening is cell membrane is being built here. Inside of a plant cell, not only would you have to build cell membrane down the middle here, but you would also have to build cell wall. Be careful with that. So this is early telophase. I got the whitefish cells. Yeah, I don't know why I couldn't find a good anaphase from whitefish. I got the onion, but it's the same idea regardless of whether it's a plant or animal cell. So we have the chromosomes are starting to unwind a little bit. We're regrowing or re-establishing the cell membrane, and you can see here there is some cleavage. In other words, we're making new cell membrane to split those cells. Here you can see the cell membrane is almost fully re-established, which means that we're almost done, and there's just a little bit of material being pinched off at the very, very end here. So this is late telophase. And then finally, the very, very last step, cytokinesis. Cyto just means cell, and kinesis means to move. So this is the cell membrane finally splits, and now our cells are called daughter cells. They are identical genetically to the cell we started with. One red and one blue in each cell. Chromatids fully unwind, and now they're back to being chromatin. And as soon as cytokinesis is done, it is back to business as usual. So if these are liver cells, they're going to do liver cell work. If they're muscle cells, they're going to do muscle cell work. So it's back to business as usual. So that's mitosis in a nutshell. Expect a video for mitosis very shortly, guys. And then we'll talk about some things that can go wrong.